Academy, and today we are going to talk about some of the important questions which came in recently in the CSIR NET Life Science 2019 question paper, and also in some of the questions related to the NEET 2019 question papers, right? So we are going to discuss all these things in this particular video. So this video is helpful for all those who are preparing for your CSIR NET Life Science exam, or for your NEET 2020 exam, or those who are preparing for your GATE life science or biotechnology exam. Please do watch this video. These questions are very important. Okay, so first question is that on sequence analysis of a double-stranded DNA, the result showed the content of cytosine was 20%. What is the amount of A and T put together? So here they are saying A and T put together means they are asking for this particular value. So as per the Chargaff rule, we know that G is equals to C, right? So that means G is also 20%, as C is 20% here, correct? So we can say G is also 20%, and we know that G plus C plus A plus T, that is the sum of all the purines and the pyrimidines in a double stranded DNA, is equals to 100%, right? So if we write the value in this, so 20 plus 20 plus A plus T is equals to 100, which gives you A plus T is equals to 60%, correct? So the option is fourth one, which is correct, 60%. So this is how you can solve these questions. Next is fruit bats known to harbor and spread several viruses that can infect other animals and humans. Which one of the following viruses is not reported to spread by fruit bats? So they have asked which among these four options has not been reported till now, which virus is not being reported till now, which is being spread by this particular bats, fruit bats. So you should remember this thing, that Ebola virus, SARS virus, Nipah virus, they all three have been reported to be spread by fruit bats, but only virus which is not being reported in these, out of these options, which is the HIV. Okay, so this is the answer, HIV. Next is the first step in the glycogen breakdown releases glucose units as. So now the options here are glucose 6-phosphate, glucose 1-phosphate, glucose and glucose 6-phosphate. So here we are talking about glycogen breakdown. That means we are talking about glycogenolysis, right? This is the process which we are talking about here, and you should remember this. In case of a glycogenolysis, the reactions occur is like this way, that glycogen, if this is the number of carbon atoms N, this will break down into glucose 1-phosphate plus glycogen N-1. Just remember this particular equation, then you will always remember the answer for this. So every time there will be a removal of a one monomer of a glucose in the form of a glucose 1-phosphate, right? So that's why 1 is removed, then N-1 will come here, glycogen. So in the first step, they are asking the first step in the glycogen breakdown releases the glucose units as glucose 1-phosphate. And you need to understand that why this glycogenolysis is so important, where it is important. Mainly they are this glycogenolysis occurs in the livers and in the muscle cells. Okay, now what is the role of this glycogen breakdown in the liver and the muscle cells, why it is required? So I would like to tell you here that in liver, the main purpose of this breakdown of the glycogen is for the release of the glucose into the bloodstream, so that it can be taken up by the other cells. All right, and in case of a muscles, this glycogen degradation is required for the giving a source of glucose 6-phosphate, which then enters into the glycolysis pathway, and this gives instant source of energy or immediate energy source for the muscle to contract. So that's why this glycogen breakdown is required in case of muscles. Next question is, a patient comes to a hospital complaining of a vomiting and a diarrhea. The doctor suggests that the patient take glucose and electrolyte solution orally. Which among the following membrane proteins likely to be involved in rehydrating the patient? Now they are saying, since obviously in case of a vomiting and diarrhea, we know there is a loss of water happens. Now they are asking out of these four options, which one is of the protein is going to involve in rehydrating that patient again. So the options are cystic fibrosis transmembrane regulator, sodium glucose transporter protein 1, insulin receptor protein, or sucrose maltose protein. So here the last option is says sucrose maltase protein. So this is basically maltase So this is an enzyme, and its main purpose is to digest certain carbohydrates such as the starch, sucrose and isomaltase, right? That is the main function of the sucrose isomaltase protein. Next is the insulin receptor protein. Now this insulin receptor protein is mainly, as the word suggests, insulin receptor. This is basically a receptor for insulin hormone, right? So the main function of this protein is involved in the regulation of the cell differentiation, the growth and the metabolism. Then next is the sodium glucose transporter protein 1. Now, as the word suggests, this is mainly involved in the transport of the glucose, and they are mainly found in the intestinal mucosa of the small intestine, right? They are mainly involved in the renal glucose reabsorption. Now, next option is the CFTR, which is the cystic fibrosis transmembrane regulator. Now, this particular protein is being coded by CFTR gene, okay, and this protein is actually present in the form of a channel in our cell membrane, okay. So this is a cell membrane, let's suppose. So this is the CFTR. It acts as a channel for the transport or certain negatively charged chloride ions in and out of the cells, okay. So it will go in and also out of the cells also, and this movement of the chloride ions in and out of the cells will lead to the control of movement of the water molecules in the tissues, okay. So in short, what it is doing, the CFTR, it is rehydrating. So this is mainly responsible, this is the protein which is likely to be involved in rehydrating the patient. So the option is CFTR. Next is, which among the following DNA markers can be used to distinguish between a homozygote and a heterozygote? Sorry, this is A and D and heterozygotes. So these are the options: RAPD, AFLP, RFLP, ISSR. So I'm not going deep into all these technologies. What are these? I'm going to take a separate topic for that, because these are very huge topics, can't be taken in this particular video, right? So just remember that for this particular question, the answer is the RFLP. RFLP stands for restriction fragment length polymorphism. So this is the molecular marker technique, okay, in which the DNA is being digested by means of a restriction enzymes, right? Since it's saying restriction fragment length polymorphism, so basically we are producing certain fragments of the DNA, and then we are getting probed to it, certain with a radio labeled or fluorescent labeled molecule probes, and finally these are and restriction fragment, because we are restricting it with the restriction enzyme, and restriction enzyme, we know they always cut at a certain specific signal sequences or recognition sequences on either side of our gene of interest. This is one of the molecular marker techniques. So the answer is the RFLP. Assuming that the A, B, C and D genes are not linked, the probability of progeny being this from the cross between this and this parents would be. So you should know how to do the crossing, okay. So let's suppose if this is one of the parent, as they have mentioned in this particular question, these are the parents with these genotypes. So let me write down here, this is one of the parent, it is being crossed with the second parent, which is this one with this particular genotype. Now you need to first find it out, what is the possible gametes which can be formed from these parents. Let's suppose if this is female and this is male, right? So let's draw a Punnett square. Always draw a Punnett square in order to study the crosses, okay. That is the easiest method. So if I draw here the female gametes over this side and the male gametes over this side, right? So what is the possible number of female gametes we can get? A, capital B, small c, capital D. Then again A, B, C and small d1. Then one more, what we'll get? A, small b, small c, capital D, A, small b, small c, small d, right? So these are the four possible type of gametes which we can get from this particular female, right? Now from the male parent, what possible number of gametes we can get? So just look into the genotypes. Obviously we can get only one type of gamete which will have a combination of alleles, A, capital B, small c and capital D. Now just you have to make the combinations, right? Possible combinations of progeny we'll get is this: capital A, small a, capital B, capital B, small c, capital D. Now in this case it will be capital A, small a, capital B, small c, capital D, small d. Now in this case it will be capital A, capital B, small c, capital D. In this case it will be capital A, small a, capital B, small c, capital D, small d. Right. So now look into the question, which the probability of the progeny with this particular genotype. So just look into our possible progeny which we get. So this is the one which they are talking about, capital A, small a, capital B, small c, capital D, small d. So any other with this genotype is not present. So the probability is 1 out of how many? 4 progeny which we get, 1, 2, 3, 4. So the answer is 1 by 4. So this is the answer for this particular question. Next, which of the following organelle does not contain DNA? Now you should know that the organelles which contain DNA are which one? One is the mitochondria that we know, and one is the chloroplast, right? So now let's look into which does not contain. So the options are lysosomes and vacuoles. Yes, lysosomes and vacuoles does not contain DNA, but let's look into other options also. So here it is mentioned chloroplast in the second option, so obviously this can't be an option. In the second one it is mitochondria, it can't be an option. Third also mitochondria is given, it can't be an option because they contain DNA. So option first is the correct. Next is respiratory quotient value of tripalmitin is. Now respiratory quotient means so basically RQ is the amount or the volume of the CO2 which is released to the volume of the oxygen consumed. So any oxidation reaction wherever there is a one kind of a substrate is getting oxidized by an organism or by an cell or by a tissue. So the RQ value, by looking at the RQ value, we can say that which type of a substrate it has used for respiration of oxidation process. For example, the protein, the RQ value is 0.9, or you can say it is in a range of 0.8, right? So 0.8 or 0.9 if it is come RQ value, that means an organism is using protein for a oxidation process. Similarly, carbohydrates have one value of RQ, right? So you can take an example of a glucose respiration where 6 molecules of the carbon dioxide is being released and 6 molecules of oxygen is consumed. So the value is 1. Now here we are talking about the tripalmitin. So tripalmitin is a triglyceride, that means we are talking about the fat, and fat we know that it has a RQ value of 0.7. So this is the option for this particular question. This next question also based on respiratory quotient value in anaerobic condition. Now this question I have placed here. This has not been come in the NEET or CSIR examination. This is just for your additional knowledge. If such kind of a question also comes, here you have been asked for respiratory quotient value of anaerobic condition. Now in earlier case we have talked about the aerobic condition. Now anaerobic condition means, since we are saying it is what? Volume of carbon dioxide released to the volume of oxygen consumed, right? So if we talk about the anaerobic condition, in the anaerobic condition the oxygen is not present, so that means the volume of oxygen consumed will be 0 here, and if we divide by the carbon dioxide amount, it will comes to infinity, right? So for this particular option you can directly click on infinity. So respiratory quotient value in anaerobic condition will always be infinity, so just remember. Next is the concept omnis cellulae a cellular regarding the cell was first proposed by. So this I hope you might be knowing. This is very easy question. This is Rudolf Virchow. Next, correct sequence of phases of cell cycle is. Now this is very important question. This most of the time it comes. Okay, so because this seems to be confusing for the students, that how the cell cycle moves, how the different phases goes. So just remember, it always starts from growth phase 1. Okay, then it will go to S, which is the synthesis phase, then it goes to the growth phase 2, G2 phase, and then it goes to the M phase, and finally G1, right? So that's how the cell cycle goes. So just remember this sequence. Alright, so by looking into this sequence you can say that the option number 2 is correct, which is G1 to S to G2 to M. Next, which of the following immune response is responsible for the rejection of the kidney graft? So rejection of the kidney graft is being done by the cell mediated immune response, CMM. Okay, remember this. Next, which of the following features of genetic code does allow bacteria to produce human insulin by RDT technology? Okay, so genetic code is nearly universal, genetic code is specific, genetic code is not ambiguous, genetic code is redundant. So you should know that the important features of the genetic code which actually allow the bacteria to produce human insulin, this particular feature which says that genetic code is nearly universal. That means the same genetic code is applicable for bacteria also, the same genetic code is applicable for humans also, for animals also, like for everywhere it is the same, it is universally applied. So here, since they have said about the bacteria, so obviously this is the correct answer. Genetic code is nearly universal. So that's it for today. Thank you so much for watching. Please do subscribe MS Bio Academy.